questions 21 and the last two, 37 and 38. Yeah. So marks are up. Marks will be up. I'm finalizing them right now. They're not up. It means you ignore Brittany. She doesn't exist now. She doesn't exist. She's just a ghost. Yeah. So no, she we will do like next class. Brittany likes to do that, and I agree with her. That's her thing. She gives. She always explains which question was problematic. From what I saw from Scantrons, it's pretty good. Like it's not bad at all. Like to me, in my opinion, there are people who did very well. There are people who did very bad, and that's quite normal. No, that's quite normal. It's so okay. Last year, I will tell you honestly. Last year, when I gave first time this quiz, I didn't have those two last questions. It was pretty much same quiz. Like nothing. Maybe I removed or added one or two questions. My average was 86%. And I actually went to Corwin to talk. Like I was invited for conversations. Like, what's wrong with you? How is that possible? Like you cannot have such a high. It's weird. Like it was too easy. That's what it was like. I was criticized because apparently when you make exams, it has to be designed so it distinguishes between people that will get B minus and people that will get A plus. Because if you don't do that, then I will try to quote, you are not maintaining the academic integrity of the institution that hired you. End of quotation. So that's my point. It has to be like that. This is my second time teaching this class. So I'm not really good on that list. I'm already better than last year. Anyway, so let's move on with the fat bacteria. That's how we will call them. And that is not official name. The reason why I'm calling them fat is by the letter of each name, because each class. Simple because I know it's hard to memorize. So I want to give you some sort of way to memorize. So we will talk today about fat bacteria and bacteroides. In next class, we will talk about something that I call CPV, or Chlamydia, Planktomyces, and Verrucomicrobia. And I will keep calling them by that abbreviation just so you can memorize it better. So let's talk about Firmicutes first. So F in the fat. Who are Firmicutes? Again, another abbreviation, lactic acid or LAB bacteria. So there are three of them that belong to Firmicutes: lactic acid bacteria, non-sporulating, and sporulating bacteria. Non-sporulating and sporulating you will memorize easy. LAB bacteria or lactic acid bacteria, it's kind of redundancy, LAB or lactic acid bacteria, but I like to call them LAB because it's easy for people who are not in microbiology to memorize. They are organisms that you probably all heard about very commonly because they are known by industrial scale production of lactic acid. They're known to be safe. Most of them have GRAS status, which means generally recognized as safe. Most of them gram positive, but not all of them. So it doesn't really matter whether they're positive or negative. Most of them are gram positive. They are using glycolysis to produce lactic acid. That's why they're called lactic acid bacteria. And they're doing that in anaerobic conditions. So we talked about them. We already mentioned them when we talked about metabolism, which is anaerobic metabolism. So they're also non-sporulating. So again, you will have later on, we'll talk about non-sporulating and sporulating Firmicutes. So but what is more characteristic of lab organisms is that they are extensive fermenters, and you will know that they belong to lab Firmicutes because they produce lactic acid. What means fermentation? That means they don't have electron transport chain, which means they don't respire, they don't have cytochromes, they don't have oxidative phosphorylation. They are just running that specific substrate level phosphorylation, and they ferment sugars. Many of them are aerotolerant, which means they can survive in presence of oxygen. They don't like, but they will survive. And again, it's a simple repetition you remember of homofermentation and heterofermentation. When we talked about metabolic diversity, you remember we talked about homofermentation when you have only lactic acid, and when you have heterofermentation, when you have ethanol, lactic acid, and carbon dioxide, and sometimes other products. So just a brief reminder. Two important genera famous in lab. Again, there are many. I'm just giving you two, and it's easy to memorize because if something is lactic acid, it will be called lactobacillus. One thing that often causes confusion is streptococcus, simple because people get confused with streptomyces. But we will talk about it, and I will tell you how we will distinguish it. So lactobacillus and streptococcus are famous lactic acid bacteria that belong to Firmicutes. And we will talk about lactobacillus now. You probably already heard a lot about this organism because it's used to prepare fermented milk products. Very common in dairy products. It's growing while fermented, so it's so good in industrial scale level. And one of the most important one is Delbrueckii or lactobacillus. Again, you will always recognize, like second word really doesn't matter so much because when you see lactobacillus, you will know it's used in preparation of dairies. Lactobacillus delbrueckii is famous in preparation of yogurt, and the reason why it's possible to grow so well while fermenting is because this organism is very highly tolerant to gluten. What does that mean? Because while it's growing, it's excreting lactic acid, which means it reduces pH of surrounding media. The organism that is not tolerating low pH will die, but this one is tolerant, so it can actually finish the final stage of fermentation because it doesn't care about low pH. To illustrate bacterial Firmicutes diversity, and streptococcus are very well representation. So we talked about lactobacillus, how it's famous in yogurt production, but there are many. Streptococcus, on the other hand, can be actually very pathogenic. It also makes lactic acid. That's why it belongs to lactic acid bacteria. But unlike lactobacillus, this one can be actually very pathogenic for humans and animals. But not all of them, as usual. Because streptococcus can also make lactic acid if it's not pathogenic. If there is non-pathogenic strain like this Thermophilus, then you will use Streptococcus thermophilus together with lactobacillus to make yogurt. And what does the name Thermophilus tell you? It's high temperature, exactly. So if something is Acidotheobacillus, means likes low pH. If something is Thermophilus, it likes high temperature. If something is ferrooxidants, means oxidized iron. So that's why you don't have to bother too much memorizing. You can just read it and know. So, and those two organisms are growing symbiotically during yogurt fermentation. And what they make in that fermentation is lactic acid and ethanol. Acetaldehyde, but acetaldehyde comes before ethanol. The point is that those two organisms are actually responsible for characteristic yogurt flavor. And Streptococcus thermophilus is usually responsible in cheese for that acidification. But even though some of them are very nice, like Thermophilus, there are those that are mean and they don't love us. They actually like to lyse our cells. And who took micro-265? You all did. Okay, so you remember Pyogenes, viridans, Streptococcus, right? You did blood. Do they still do blood agar? Yeah. So, did you hear about hemolysis? Yeah. So, Streptococcus, the bad one, Pyogenes and viridans, they can either strep throat or dental caries, depending on the organism, but they're categorized based on how they perform hemolysis on blood agar. So they can either be hemolytic or not. And we will go through that. I have a mini reminder here for people who don't have microbiology experience. Again, I apologize for people who already heard this. There are people who never heard about this and they will benefit from repetition. What is the characteristic of streptococcus? They produce a specific type of hemolysin or streptolysin, which can be the streptolysin O or streptolysin S. If it's O, that means it's oxygen sensitive. If it's S, it means it's stable with oxygen. Now, if it's streptolized in S, then based on the way how it performs hemolysis, they are categorized in complete and incomplete. And then incomplete can be alpha and gamma. We will talk about that now. So briefly, so they make streptolizin. That's same like streptolizin is type of hemolysin. So that means it lyses or it acts on red blood cells. Now, either can be complete or incomplete beta hemolysis. So how is that characterized? So if it's complete beta hemolysis, so-called pyogenes group. So Streptococcus pyogenes does complete beta hemolysis. And that's how they're called, pyogenes group. If they have that, if their streptolizing has the ability to lyse blood cells, this is what you will see on blood agars. So you see these round-shaped colonies with a white surface, and you see zones of hemolysis. Like hemolysis means lysis of blood cells. So that's complete beta hemolysis. White one. If you see viridan, if you have viridans group, and this is in past, this was very common metabolic test done in hospital setups, just to distinguish between two types of streptococcus. If you have incomplete beta hemolysis, then it's a viridans group, and it's an alpha or gamma, and you will see green agar or this whitish one. Beta would mean it's broken, cell is broken. Let's see how that goes. So alpha and gamma are both classified as incomplete. Beta is complete. And again, just additional information if lacking microbiology experience. I was told that many people did not take micro 265 here. So how the hemolysis functions? So bacterium, Streptococcus viridans or pyogenes, is secreting certain toxins in media, which are called hemolysins or streptolysins. So streptolysins or S, in this case, S is a type of hemolysin. But because it's from Streptococcus, it's called streptolysins. So those hemolysins or streptolysins, they perform hemolysis, which means a lysis of red blood cells. And depending on how that lysis happens, you see the changes on the blood on the agar. Again, it's not only Streptococcus. That's why there are other organisms that produce hemolysin. That's why this one from Streptococcus is called streptolysin. So what's happening in incomplete hemolysis, which is alpha hemolysis? So what it happens here, so you will see green or gray-brown columns on the plate. So when you grow it on the plate, you say, okay, this is incomplete alpha hemolysis. What's the mechanism? So bacterium, Streptococcus, makes hydrogen peroxide. And what hydrogen peroxide does, it converts hemoglobin from blood to methemoglobin, which causes the green color. But cell membrane of the red blood cell remains intact. That's the point. And that's Streptococcus pneumoniae. It is, if you have beta hemolysis, that's dying, killing a cell. Well, the other thing is the same, but this is, so if you have complete hemolysis, streptolysins, specific type of streptolysins, streptolysins S, will interact with the cholesterol in cell membrane of your red blood cells, and it will form pores. And that's why you get like a completely breakage of the cell. That means you will see clearance of the membrane, like you will see completely lysed cells. It's very characteristic. In gamma hemolysis, again, it's incomplete. Alpha is also incomplete, but in alpha, you will have that change from like to greenish color because hemoglobin gets converted to methemoglobin by hydrogen peroxide. Gamma is not. There is no effect. So there is no hemolysis. And that's Neisseria meningitis Enterococcus fecalis. So I'm pretty sure that in micro 265 you have Enterococcus fecalis that does not perform hemolysis, and then you probably get plates where you have to distinguish which strain is. So they give you a plate with Streptococcus that does hemolysis, pyogenes, and give you with Enterococcus, and then you have to decide. You have to write which one does. Again, people who took it probably know, but there are people who didn't. So maybe this will be good motivation. So these are all from lab bacteria, Lactobacillus streptococcus. Non-sporulating bacteria, so other than these two, because the lab are also non-sporulating, are pathogens, Listeria and Staphylococcus. So you probably had Staphylococcus epidermidis in Micro 265, which is the one that lives on our skin. Listeria, I'm pretty sure you didn't have because it's highly pathogenic. But it has some interesting features that I am going to share with you in a few minutes. So, again, only two genera comparing to many others that are present out there. So, what is Listeria? Who heard about Listeria monocytogenes? Yeah, so that one can actually be a very big problem for pregnant women. So the first time when I faced with that organism was when my lab mate in Wyoming was pregnant, and I was doing her experiments because she couldn't enter the lab. So it's so pathogenic that literally you are not allowed to enter the lab. Like, like it's only specific lab where you work with that organism. Like my friend was not able to enter the lab because she was pregnant. It's really, really bad. But as usual, microbiologists learn how to exploit interesting features of the worst possible, the most evil microbes in the world. And that's using the Listeria as a cancer immunotherapy. So this citation is taken from a paper that should be in your notes. But the bottom line is that Listeria, for some reason, Listeria really likes to live in tumors. It really likes anaerobic environment, and Listeria's ability and love to live in tumor area was used to generate cancer immunotherapy and cancer vaccines. So how they did that? So I found that very interesting, and although there is a side note that I will tell you later on. So they use attenuated, like disabled or like attenuated or disabled Listeria strains. What means attenuated means they delete virulent genes responsible for virulence, for pathogenicity from virulence. And then they fuse this Listeria antigen they fuse Listeria, they use Listeria antigens with some adjuvants to enhance anti-tumor response. And the reason why that is because Listeria really likes to live in tumor cells, so you would fuse specific anti-tumor factor to Listeria antigens, and then you would target specifically tumor tissue. Because as you probably heard, one of the big problems in cancers, in chemotherapy, is that when you do treatment, it's not very selective. It treats all of your organism, but people are thinking that Listeria can actually can be specifically directed to tumor tissues, and it can. Side note of that, again, this is not testable, just for your, just FYI. I had a chance to see how engineering of Listeria goes. So for some reason, Listeria really doesn't like to be engineered. And so people in the lab where I used to work tried to modify Listeria to be light controlled, which means you would direct it to tumor and you would keep it low, like low activity, until you shine light. And then you shine light, and then Listeria starts delivering drugs. The problem with that, why would you do that? Because you want to do control with light instead of chemicals, because chemicals are diffusible nature. The problem with that is in order to make something light responsive, you have to have specific molecules that would absorb that light. It's called chromophore. This baby doesn't want to get chromophore in. It just doesn't, it just cures out. Like you put plasmid, it cures it out. So I know a person who spent 8 years working with that and nothing happened. So I'm not saying maybe it works now, but I would not dare doing that. I really want to live a long life. Anyway, another one would be Staphylo. Epidermidis non-pathogenic, found on skin. Epidermis, skin. Aureus, pathogenic. Very, very common in osteomyelitis, meningitis. You would see it in pneumonia. It's very present in hospitals. Next one, sporulating bacteria. So now this one is completely different than lactic acid and non-sporulating. So there are two of them that are called Bacillus and Clostridium. So you probably heard of all of them, just you never knew that they belong to Firmicutes. And both of them have some specific characteristics, especially Bacillus. So, what is characteristic about these bacteria? They form endospores. Endospores are usually characteristic of fungi, but these bacteria actually are very good in forming spores. So, in the spore formation is a very big advantage in soil. Why in soil? Because soil is such a dynamic and very exposed environment, and what can happen is very often you have like a change in environmental conditions, and then microorganism runs into unpleasant conditions, and then it has to form endospore. If there is no ability for endospore, microbes die. But those that have ability to form endospores, they can actually survive because the purpose of endospore formation is to preserve DNA, so later on organism can propagate. Just FYI, again, non-testable, some bacillus spores are found from like billion years ago. Like they're really, really well preserved. They help, and then the spores help him survive. But I'm not going to talk about that was preserved in endospores. I will talk about Bacillus thuringiensis, which is very easy to memorize. And if you ever heard about GMO plants, then did you hear about BT maize? Like GMO plant? Yeah. So what is done with Bacillus thuringiensis is causing fatal disease in insects. Kills insects. Now, the way how it does is because it makes different toxins that target different groups of insects. So this is called BT toxin. It's very famous BT toxin. So that gene for BT toxin is introduced into the soybeans, maize to make plants, GMO plants that are insect resistant. And that's very famous, especially in US. So those GMO plants are quite diverse. They make them, and that's done from using a toxin from Bacillus thuringiensis. Clostridium is another sporulating bacteria that is also fermenter. So it does not have respiration, it has absolute level phosphorylation. It performs sugar fermentation. And what is specific about this organism is presence of celluloses. So these black dots are called cellulosums. What does that tell you? What would cellulosum have? Enzymes to degrade cellulose. So why is that good? Those cellulosums are called the nanomachines. That's how, like, no, not nanomachines, molecular knife, And I teach about that in deep in 315 when we talk about biofuels. Why? Because celluloses are able to bind insoluble cellulose and degrade it into the soluble products, which would be glucose, and then those glucoses are transported back to cell to be degraded in the bacterial organism. Why is that good? It's good because when we talk about biofuels production, you want to get cheap carbon source. So to get cheap carbon source, of course, you will not take glucose, like pure crystalline glucose or table sugar. First, food versus fuel debate. Second of all, really expensive. So you want something that is waste. What is waste? Lignocellulosic biomass. What is the problem with lignocellulosic biomass? It has cellulose, hemicellulose, lignin. Cellulose is polymer of glucose, right? But it's very hard to degrade. So with these guys, cellulosums, you can actually break down cellulose, and then this is usually used in production of second generation of biofuels. And again, I talk more about that in Micro 315. This is only for but FYI. Again, Clostridium butyricum and aceticum. Easy to memorize. Why? What is butyricum doing? Produces butyric acid. What is aceticum doing? Producing acetic acid, which is vinegar. There is literally not too much to memorize, just to understand what means acetic acid, what means butyric acid. And both of them are value-added compounds. The Clostridiums, especially botulinum and tetany, are and perfringens, they're pathogenic. Perfringens is causing gangrene, which is really bad disease, and you can lose a part of your body. Botulism, tetanus, you've probably heard about that. Clostridium botulinum, you can imagine where we use it. Who likes Botox? So I'm 38 years old, so I need it. I have to be fresh. Although first you look like bees attack you, but it's fine. You don't get out. You just sit at home while you heal. Not that I know, but I will know. Anyway, next one is A in film. So that's how I'm talking. Firmicutes, so FAT, fat bacteria. So Firmicutes, next one is actinobacteria, and then we will cover the last. Actinobacteria are famous Mycobacterium tuberculosis. What this organism causes? Tuberculosis, exactly. So there is really tuberculosis was a big issue in non-developed countries in past. I was personally, when I moved to US, they usually think that the moment when you come from Serbia, you carry a tons of disease. So they put us in quarantine because they thought we have tuberculosis. But we were sitting 2 weeks. It was bad. So they're usually, so mycobacteria are not, usually they're not, except tuberculosis, Mycobacterium tuberculosis. The other representative of these guys this is confusion. So you remember how we talked about streptococcus? This is streptomyces. So the way how I memorized it is because when something is actin, I would think about like a filament, like actin. And because this is like streptomyces, like mycelia, and I associate mycelia with actin. That's how I memorized. And again, that's just my analogy. That's how I studied. You can choose whatever you want. But streptomyces is full organism. It's not pathogenic, not all of them, but the one that I will insist on you to know has actually a very funny story behind, and it's antibiotic producer. That's why streptomyces is very interesting in biotech, because it's a microbial palette for antibiotic production. I love that organism. And the last one is propionic acid bacteria. So like tuberculosis, streptomyces, and propionic acid bacteria. So what is the landmark of Mycobacterium? Very specific. There are different secretion systems in bacteria, and if anyone took my microbial physiology class, I teach in details. There is one type of secretion system that is very specific and it exists in Mycobacterium tuberculosis. It's type 7 secretion system, and it's very specialized for these hydrophobic cell walls. So it's very highly hydrophobic because it has this characteristic mycolic acid, which is linked to these peptidoglycans in cell wall. It's a gram-positive. Well, technically it does look like gram-positive, but it's not very, you can't really stain it very well because it's so hydrophobic. So because of that mycolic acid. So I wouldn't even characterize it as a gram-positive. In past, we were calling it gram-positive, but now we say it doesn't stain very well as with gram staining because of that mycolic acid. But it's definitely not gram-negative, that's for sure. Streptomyces is awesome. Filamentous, and it doesn't like, they have spores called conidias, but what is different is that those conidias they are spores, but they're different than those spores that you see in Bacillus and Clostridium. The most important for streptomyces is that it's actually famous antibiotic producer. And this is where the funny story comes into the game. So, Magic Healing Graveyard. That's how the story was called, Magic Healing Grave. So if my grandma was alive, she would believe in this story, I'm pretty sure. And she would think I am devil's daughter because I don't believe in this one. So what happened this was in Perth, like there was that village in Ireland. The priest was on his deathbed, he was claiming that the ground, the soil from his grave will have magic properties. So he died, they begged him, and then people would go, and the locals actually, locals would would go to his grave and take a soil, and then they would use that to heal wounds and like to sick people, giving it to like making a teeth for sick people, not teeth. I think they're putting it on wounds, but they would literally bring a soil, and they would think that's like because he was a priest, that's a magic soil from his grave. What happened later on, you can imagine. There was a scientist who came and got the soil from a grave, went to the lab, bring the soil from the grave to his team of researchers, and found that when he did enrichment, like isolated specific organisms from that magic soil, he found new Streptomyces bacteria that actually produced new antibiotics, and that's why that grave was magic. And this should be antibiotic, not antibody, sorry. So unknown Streptomyces bacteria was the one that actually healed, not magic priest. So again, that's the funny story, and that's how you will never forget Streptomyces. So PEB bacteria, propionic acid bacteria, again, PEB is not official name. PEB is how I call it, so you guys can have easier time memorizing. PEB is discovered in Swiss cheese. So it is, so how we talked about Streptococcus and Lactobacillus as a part that are doing fermentation for dairy. So Streptococcus and Lactobacillus will make lactic acid, and propionic acid bacteria will use that lactic acid as a substrate, and then will give that acidic flavor to cheese. So three bacteria, like Lactobacillus, Streptococcus, and propionic acid bacteria. So propionic acid bacteria would be what? Secondary ferment. Next one, so now we are at the T part, which is Tenery cutis. Again, we are at fat bacteria. Tenery cutis, again, you probably heard about them because they're very well known. Like if you ever went to test for a bunch of unpleasant things, mycoplasmas would be also analyzed along with chlamydia. So often they are called mycoplasmas because the notable genome is mycoplasma, that's a human pathogen, but they do not stain gram-positive because they do not have cell wall. So this is very unique in these organisms. They are bacteria, but no cell wall. They are very small. We say they resemble protoplasts, like a cell without cell wall. So that's their characteristic. And the famous genera are Mycoplasma and Spiroplasma. Mycoplasma is something that you will usually see, but if you ever go to test for chlamydia, they will give you test on Mycoplasmas and Ureaplasmas because they usually operate together. When we say it resembles protoplast, means structure that remains after you remove bacterial cell wall. This one does not have cell wall by default. But it's very resistant, surprisingly, even though there is no cell wall, it's very resistant to osmotic lysis. Why? Because it lost the ability to make cell wall, it evolved the ability to make stable cell membranes. It has sterols, and sterols, bacterial sterols, are giving the membrane that stability, so it's very resistant to osmotic lysis. Another characteristics of membrane is lipoglycans. So they're very specific polysaccharides in membrane, which stabilize that cell membrane, so it helps in attachment of mycoplasma to animal cells because it's a pathogen. So lipoglycans and sterols are what is different in these cell membranes comparing to other bacteria that have cell wall. These guys do not have cell wall, but they're still very stable. And the last but not least are bacteroides. So this, in my opinion, is very easy to memorize because they're very much involved in our gut diet. So they're easy. They could be good, but they could be bad, like every organism. So they're non-commensal in animals, and they're dominant bacteria in human large intestines. So they can sometimes become pathogenic and be associated with bacteremia when you found bacteria in blood. That's just because they naturally live in our gut. So when something happens in our organism, these guys can just go wild. Often they can produce enzymes that are not encoded by human genome, but that's why they have, that's why we have that. You probably heard about microbiomes. So microbiomes are very big deal in research nowadays. It's very significant and for our health and for literally everything. And to like Brittany is very experienced in microbiome analysis because that's what she's doing, just not in us. You're doing it in crabs, which you will in soil. Like when she gives you your guest lecture, you will talk a lot about microbiomes. But human microbiome project was a really big discovery, and it's very well funded because it was discovered that it's quite significant for our disease, for our digestion. And the reason why bacteroides are very important is because they increase they increase availability of plant polymers that we can use because they add up additional enzymes to our digestive tract, so we can digest different plant polymers. What is characteristic of bacteroides? Characteristic is that they have very different lipids. They have sphingolipids. So sphingolipids are similar lipids. Those type of lipids exist in human brain. And what is characteristic is that you will see no glycerol in lipid backbones. You will see something called sphingosine. Again, this is you see how there is a connection here. So even though it's bacteria, it does have some similarities. So it's quite different, sphingosine instead of glycerol. The last slide for today is the fun fact about bacteroides. And I really like, same how I like that story about healing grave, I really like this story about bacteroides and firmicutes. Why? So I don't know, do you have a low or high fiber diet? I am very bad because I have zero fibers in my diet. I eat all four possible things and I drink a lot of sugar. So I will probably have the bad life. But in low fiber diet, which I have, you will have more bacteroides than firmicutes. So it is all about the balance between bacteroides and firmicutes. So when you eat like Dr. Lazic eats, like nothing good, all unhealthy stuff, then you get more bacteroides and less firmicutes. What that happens is now those overloaded microbes will eat your intestinal mucosa, which means you develop ulcerative colitis. I'm pretty sure I had that. What that causes is the reduction of butyrate level or reduction of short chain fatty acids. So butyrate is short chain fatty acid. So when butyrate level drops after bacteroides eat your intestinal mucosa, that low butyrate level will affect G protein signaling, which is like a human signaling. And the G protein signaling is involved with histone deacetylase. And histone acetylation, so histones are proteins, histones are proteins that are involved in DNA packing in eukaryotic cells. So nothing for you to know. But histone modifications, acetylation, deacetylation, actually is involved in cancer progression. So that's why when you disturb butyrate level, you disturb histone acetylation, deacetylation, you die. I'm kidding, you don't die. But that, like, the point of me telling you this is just to understand how microbiome in our gut is going to be important for our health. And because it can, honestly, for me, it would be worse if I'm fat than if I have cancer. So I'm guessing that would be same for many other people. So again, this disturbed signaling will be very problematic if your bacteroides eat your intestinal mucosa. And that would be all for today. I thought that there will be short lectures, so you are free to go and to wait for Brittany and me to